This is a video for topic 2.7 in the IB Biology course. Here is all the understandings, applications and skills that you need to know. So it's quite a large topic, but it's all about transcription, synthesis and translation of DNA. Okay, so here you can see the basic method of DNA replication, where an original template is separated into two, and then a new strand is built onto that. So this theory is called semi-conservative. It was confirmed by Meselson and Stahl, but prior to this there are a couple of other methods that were proposed of DNA replication. Conservative being the original strand stays and there is a completely new strand made each time. Semi-conservative, which is the right one, means there's one strand that's from the original and one strand that's new in every replication, and then dispersive, where random bits are dispersed. So Meselson and Stahl were able to experimentally test the validity of these three models using radioactive isotopes of nitrogen. So nitrogen is a key component of DNA, so in the nitrogenous bases, and it can exist as a heavier 15 or 14N. So DNA molecules were prepared using the heavier 15 and then introduced to replicate in the presence of the lighter 14. The DNA samples were separated so you could determine the composition of the DNA in the replicated molecules. So after one division, the DNA molecules were found to contain a mix, disproving the conservative theory. After two divisions, some molecules were found to consist solely of 14, disproving the dispersive model. So here's what was seen after the first reaction: a mix, and a second replication, some 14 only strands and some mix. Up here is the method of replication run by different proteins. So helicase goes down the chain to the molecule, unlocking or unzipping the double helix, and then DNA polymerase enzymes attach free nucleotides onto the strand, matching the base pairs that are already on that original strand. This strand up here, so this area here is called a replication fork, and this DNA polymerase here that goes in towards the replication fork is called the leading strand, and that can do that much easier because it doesn't have to stop. This one here, however, is called the lagging strand, and it must synthesize the DNA in small fragments along as the DNA is being unwound. Those fragments are called Okazaki fragments, and then they're bonded together afterward. And that's because the DNA replication has to be done in a 3' to 5' direction. So the polymerase chain reaction is an artificial method of replicating DNA. So each reaction doubles the amount of DNA, and so in about 30 cycles it can create over 1 billion copies. So the reaction occurs in a thermal cycler and uses variations in temperature to control which stage of the replication is going on. So denaturation separates the two strands. Annealing is when the sample is cooled to allow the primers to anneal. So the primers designate the sequence to be copied, and elongation, the sample is heated to the optimal temperature for heat-tolerant polymerase, the Taq, to function. So Taq polymerase is an enzyme located isolated from the thermophilic bacterium Thermus aquaticus, so that lives near thermal vents in the Earth's surface, and this enzyme's optimal temperature is 75 degrees, meaning it can be used at high temperatures in PCR without denaturing. Taq polymerase extends down the nucleotide chain from the primers, therefore primers are used to select the sequence that should be copied. So here you've got your template DNA with your wanted gene. You then replicate each many times over, and the amount of DNA doubles every single time. And so you've got your target gene replicated many times with each cycle. Transcription is the process by which an RNA sequence is produced from a DNA template. So RNA polymerase separates the DNA strands and synthesizes a complementary RNA copy from one of the DNA strands. When the DNA strands are separated, ribonucleoside triphosphates align opposite their exposed complementary base partners. RNA polymerase removes the additional phosphate groups and uses the energy from this cleavage to covalently join the nucleotide to the growing sequence. Once the RNA sequence has been synthesized, RNA polymerase detaches from the DNA molecule and the double helix reforms. So you can see all of that there in that diagram. The sequence of DNA that is transcribed into RNA is called a gene. So the strand that is transcribed is called the antisense strand and is complementary to the RNA sequence. So the strand that is not transcribed is called the sense strand, and that is identical to the RNA strand, except that T would be used instead of U. The transcription of genes occurs in the nucleus, where the DNA is, before the RNA moves into the cytoplasm for translation. Codons are 3 base pairs, so the base sequence of an mRNA molecule encodes for the production of a specific polypeptide. The mRNA sequence is read by the ribosome in the triplet of bases called codons. Each codon codes for one specific amino acid with a polypeptide chain. So you can use a key like this, or sometimes you'll see it in a table. So here the first letter is the first letter of the codon, so G, A, C, for example, would give you asparagine, or UCA would give you serine, and then there are also stop codons like UGA, which would give you a stop codon. Translation is the process of protein synthesis in which the genetic information encoded in our mRNA is translated into a sequence of amino acids on a polypeptide chain. Ribosomes bond to the mRNA in the cytoplasm, and then they move along the molecule in a 5' to 3' direction until it reaches the start codon of AUG. Anticodons on tRNA molecules, so this is a tRNA molecule, pair with the appropriate codon shown here. Each tRNA molecule carries a specific amino acid on top, so that on top, which allows for that codon on the mRNA to directly influence which amino acid joins onto the polypeptide chain. Ribosomes catalyze the formation of peptide bonds between the adjacent amino acids, and the ribosome moves along the mRNA molecule, synthesizing the polypeptide chain until it reaches a stop codon. At this point, the translation stops and the polypeptide chain is released. So genetic code is often referred to as universal because almost every living organism uses the same code. There are a few rare exceptions, but you don't need to know about them. So one of the benefits of this is that genes can be transferred from different organisms because they are the same gene, and different organisms can read the gene. So for example, in humans there's a gene for producing insulin. That insulin gene can be combined with a plasmid from a bacteria, which can then be put back into the bacteria and grown in culture, that specific bacteria, which means that bacteria is producing a lot of insulin, providing us with a very fast and efficient way to create insulin by creating this recombinant DNA. But that is replication, transcription, and translation, topic 2.7 for IB biology.